Hi guys, welcome to our channel. This is the Science Lectures. Please, thanks for coming to our channel. Consider subscribing and like our videos, please. And you comment in the comment section. So, we are in our series of cell organelles, eukaryotic cell organelles, and we looked at the nucleus earlier on. We saw also the rough and smooth endoplasmic reticulum. We are currently going to look at the Golgi apparatus or Golgi body. So we are going to start off with our Golgi body. Golgi body, or you can call it the Golgi apparatus, or we can call it the post office of the cell. Now the post office of the cell, its major function or major role is receiving contents. Let's say this is our rough endoplasmic reticulum, and this is our smooth endoplasmic reticulum. They produce transport vesicles. Let's say this is a vesicle, transport vesicle, and this one is also a vesicle from the rough endoplasmic reticulum. The Golgi body will receive vesicles produced by the rough endoplasmic reticulum and vesicles received by the smooth endoplasmic reticulum. So that is one basic function of the Golgi body, receiving contents of vesicles from the rough endoplasmic reticulum and from the smooth endoplasmic reticulum. Again, what we need to know about the Golgi body, it is containing flattened sacs known as cisternae. Cisternae are these flattened sacs of the Golgi body. Then the face that is next to the rough and smooth endoplasmic reticulum that receives the contents of the rough and smooth endoplasmic reticulum is the cis face. The cis face. CIS face. Then the trans face is the one that is further away from the rough and the smooth endoplasmic reticulum. This is the trans face. So you can decide to just say CIT, the cis phase, the middle phase, which is the intermediate phase of the Golgi apparatus, then the T, which is the trans phase of the Golgi apparatus or Golgi body, or the post office of the cell. So what is very important with this Golgi body? First of all, the Golgi body, we can call them, I think we can go straight away to the functions, or we can elaborate more. Functions of the Golgi body. Number one function is receiving contents of vesicles. It receives receiving vesicles from the endoplasmic reticulum, the ER. That is both the rough and the smooth endoplasmic reticulum. So it receives these vesicles from the rough and the smooth endoplasmic reticulum. What happens after receiving the vesicles? We receive these vesicles and we'll do what we call modification. Modification of the vesicles. It is going to modify the vesicles. What is involved in vesicle modification? Vesicle modification, this is what is involved in vesicle modification. Remember, the contents it is receiving are from the rough endoplasmic reticulum. And we very well know that in the rough endoplasmic reticulum, let's say this is our rough endoplasmic reticulum. The lumen, let's say this is the lumen of the rough endoplasmic reticulum. In the lumen of the rough endoplasmic reticulum, we say a protein, messenger RNA, comes in with ribosomes. And when it comes in with ribosomes, the rough endoplasmic reticulum will do protein synthesis. So it forms a protein. Let's say in the lumen, we are currently having a protein. So we say if this protein is present, it will undergo what we call, they will add a sugar to the protein. Let's say this is the protein. We activate it with a sugar. This is our protein. We add a sugar. A short sugar chain, which is the oligosaccharide. We add an oligosaccharide. So when we add the oligosaccharide, what happens? Protein folding, we say the rough endoplasmic reticulum also does protein folding. So we fold this protein with its sugars into the 3D structure. We fold it into the 3-dimensional structure. So after folding into the 3-dimensional structure, what happens then? We package it into a vesicle. After packaging it, we release the vesicle, which vesicle will go to the Golgi body. So why is this all important? First of all, a brief recap on the rough endoplasmic reticulum. We said number one, protein synthesis. We synthesize the protein. Then we add a sugar, what we call activation, or we can have glycosylation of this sugar of the protein. We glycosylate the protein. Then we fold into the 3-dimensional structure. We package it into a vesicle. We release the transport vesicle to the Golgi apparatus or Golgi body. So what are the modifications? We are now right back to the Golgi body. What are the modifications that will happen? There will be cleavage of we shall cleave off this oligosaccharide after receiving what was brought by the rough endoplasmic reticulum. So cleavage, modification, one of the modifications is cleavage of, they can ask a question, modifications that happened, modifications in the Golgi apparatus. So this is the number one modification. We can have cleavage of the oligosaccharide. We can cleave off the oligosaccharide chain. So cleavage of this oligosaccharide, we cut off the oligosaccharide. Then which other modification can happen here is addition of sugar moieties. So we can add some sugar, addition of sugar moieties. So we can add some sugars, some sugar moieties. Then what else can we do? We can phosphorylate. We can decide to add phosphate groups. We can add phosphates, phosphate addition or phosphorylation, just to activate or to mark. Okay, it acts as a marker to the protein. We can do phosphorylation. We can add fatty acids, fatty acid addition. So we can also add the fatty acids. Then, most importantly, we can also, under the modification, we can also removal of the monosaccharides. So we can cleave, we can remove the monosaccharides. Removal the monosaccharides. So these are the modifications that are carried out within the Golgi apparatus, or what we call the Golgi body. So it will receive the vesicles, but on receiving the vesicles, it can modify them in this way. But the Golgi apparatus can as well add markers. It can add markers. It can decide to add markers. We can add markers. Markers, or what we can refer to as identifiers. We add markers such that we can easily locate the destination, to provide a destination for these molecules after addition of the markers. So we can add markers or tags or identifiers that will direct the molecule to its site of action. Interestingly, also the Golgi apparatus can synthesize lysosomes. Which lysosomes are involved, contain most of the hydrolytic enzymes. And these hydrolytic enzymes within the lysosomes are already synthesized in the rough endoplasmic reticulum. That one is important for us to note. Then what happens next? What else can the Golgi body do? This Golgi body can now package these molecules and transport them to their final destination. Package them and transport to their final destination. And the destination can either, these proteins synthesized can either be excretory proteins which are going to move out of the cell via the cell membrane. Then they can as well go to the cell membrane and other cell organelles, cell membrane and other organelles And finally, these proteins can also be lysosomes, which lysosomes are very important in breaking down organelles, in breaking down different components within our cytoplasm. So that is it about the Golgi apparatus. So a brief about the Golgi apparatus, a recap. We have different cisternae, the piles that are known as cisternae. We have the side that receives the vesicles from the rough and the smooth endoplasmic reticulum. The ones we refer to as the cis phase. We have the trans phase that is further away from the rough and the smooth endoplasmic reticulum. Then it is receiving the transport vesicles. Once these vesicles are received, they can be modified. And in the modification process, we can have cleavage of the oligosaccharide chain, which oligosaccharide was added when the molecule was still made in the rough endoplasmic reticulum. We can add some sugars to the molecule during modification. We can add phosphates, we can add fatty acids, we can remove some monosaccharides, and we can do the addition of markers and tagging. Then it can synthesize lysosomes, excretory proteins. It can as well synthesize the contents of the cell membrane. In our series of the cell organelles of the eukaryotic cell, we are still continuing with the cell organelles. You know, there are many cell organelles within our cell, so we still have to continue. But other videos where we're talking about the nucleus, the rough endoplasmic reticulum, the Golgi body are available on our channel. You please check them out. So today we are going to look at the interesting lysosomes. They are very interesting. So we need to know what are lysosomes. But first of all, when we break down this word, we can get a word lysis inside there. Then we have also a word some, which means a body or a cell. So lysis just means, okay, breaking down, splitting. We can split something, we can break down something. So that is lysis. So basically our lysosomes will deal with splitting, breaking down. That is what they will do. From the basic principles, lysis, that is what lysis is all about. So what is the importance of these lysosomes in our cell? First of all, these lysosomes are manufactured or synthesized by, guess who? The Golgi body or Golgi apparatus. The Golgi body. In our series of the Golgi body, we evidence this. The Golgi body is capable of synthesizing the lysosomes, which are very important in our cell. Now, lysosomes are membranous organelles. Let's say they have membranes. Let's say this is the membrane of our lysosome, but inside the membrane we have enzymes. Inside this membrane we have enzymes, and these enzymes are referred to as, we can call them hydrolytic enzymes. Hydrolytic enzymes inside the lysosomes. Hydrolytic enzymes inside there. But these enzymes work at a pH which is acidic. They work best at the acidic pH. A pH of around 5 is favorable for them to do their hydrolytic roles, these hydrolytic enzymes. Examples of such hydrolytic enzymes, examples of these enzymes, we can have enzymes like nucleases, which will break down nucleic acids. We have lipases. Of course, these ones are breaking down lipids to their simpler molecules. Then we have also enzymes. We can have also acid phosphatases. Acid phosphatases. Acid phosphatases, and a vast majority of other enzymes that will break down contents. So basically, hydrolytic enzymes inside there work best at a pH of 5, that's an acidic pH. Then we have enzymes like nucleases, lipases, acid phosphatases, glucosidases. All these enzymes will work in breaking down the contents inside the cell. So what are we interested in? We are interested in the functions of this lysosome. Functions of the lysosome. Functions of the lysosome. Number one function of the lysosome is to break down. Let's say we have a white blood cell. White blood cell. White blood cell engulfs a bacteria. It engulfs a bacteria or a virus. So this bacteria or virus will be broken down with the aid of the lysosomes. So number one role is breaking down of microbes. You can break down microbes. Let's say we have a virus, or let's say we have a bacteria inside our cell, so it will be broken down with the aid of the lysosome. Then let's say we have a food material entering the cell. Food material entering into the cell. We need to break down this food material. The food material will be broken down also by the lysosome. So breakdown of food materials. Any food materials will be broken down. Let's say inside the cell we have accumulation. If this is our cell, and we have accumulation of waste materials, waste particles. So breakdown of those wastes. Waste breakdown. So we can break down the wastes to more safer compounds. Waste breakdown. Break down those wastes and release out the rest of the contents. Then what else? Let's imagine that our cell has worn out cell organelles. Let's say the mitochondria in our cell is worn out, it is growing old, and it is no longer functioning. The cell will break down the worn out cell organelles, and that is what we call autophagy. 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 This is breaking down the worn out cell organelles. Worn out cell organelles. They can no longer carry out their function. So what we do is the lysosomes will break down these worn out cell organelles, and when they break down the worn out cell organelles, the raw materials released can be used to synthesize other cell organelles. That is what we call autophagy. But again, we can have a situation where the whole cell is no longer functioning, where the whole cell is no longer important, is no longer working very well, is growing old. So we can have the whole cell destroyed, and this is what we call autolysis. Autolysis or autolysis. So autolysis is where we break down the entire cell. So this is it basically for the lysosome. We said lysosomes, lysis, splitting, breaking down. They are synthesized from the Golgi apparatus, and they contain hydrolytic enzymes. However, they are covered by the membrane, system. Because if these enzymes were just exposed to the exterior, they can digest the organelles, the entire organelles of the cell. So the membrane is basically for that. Then they work best at a pH of 5, which is an acidic pH. Examples of such enzymes we have nucleases, lipases, glucosidases, acid phosphatases. Then the functions, we can break down the microbes, the food materials, can break down wastes, can do autophagy where we break down the worn out cell organelles. We can do autolysis where we destroy the entire cell. So that is it for the lysosome.